Okay, so this is a revision of chapter 19, which is the blood. We already went through everything in this chapter, but let's focus more on what is the core of the chapter, what's more important. So the first thing will be functions of the blood. This is something that you need to remember, generally speaking. Okay. I'm not going to re-lecture again. I'm just pointing out to the more important stuff. So we know the difference between plasma and serum. This is obviously important. The function of each one of the cells, generally speaking, is important. Hemopoiesis, the mother cell and stem cells, this is important. There are some numbers that we need to focus on as we go. You can put them together, you can do whatever you want. There's like 6, 7 numbers in the whole chapter that you need to remember. Specifically, the temperature and the pH. You have to remember these numbers, and we'll give you numbers as we go. Blood volume in liters is important, males and females, and the rule is males always more than females, any number in the blood. So the plasma, what's the difference between extracellular and intracellular? Okay, this is important to remember. Extracellular and intracellular. What's the difference? Which one has more oxygen, carbon dioxide, and plasma proteins? So this is one. Okay, plasma proteins, you need to remember the main function of each one of the 3. Albumin, globulin, fibrinogen. Right? One word. Albumin, transport. Globulin, antibodies or immunoglobulin, and then the transport globulin as well, but it's more important the antibodies or immunoglobulins. Fibrinogen, blood clot. It will become fibrin and help in the blood clot. Okay? Where do we make our plasma proteins? Most of it is in the liver. There are 3 ways, 3, to know if your red blood cell is normal or not. Red blood cell count, this is 1. Hematocrit, you need to remember what hematocrit means. So it's not only the number, hematocrit. Here is a number that you need to remember. So these numbers are also important. What's a number? The normal number and the normal hematocrit. Okay? We should understand what hematocrit means, generally speaking. And the normal number of the count and the hematocrit. Lifespan, important, 4 months. The normal hemoglobin in males, 14 to 18. In females, it's less. So it can be like 13 to 16 or something, but just remember this number, that will be good enough. Hemoglobin. So obviously, that will get us to anemia. What's the anemia? Any of the 3. Low blood count, low hematocrit, and low hemoglobin concentration, right? What's oxy and deoxy and carbaminohemoglobin? What each one of the 3 means? Talked about anemia. Those 2 terms are important, and you will see those. I think it will be in the concepts. So what's hemoglobinuria and the difference between hemoglobinuria and hematuria? Jaundice. What's jaundice? Okay, this is important, which is increased bilirubin for some reason. Yellowish discoloration due to increased bilirubin. So the first type is the red blood cells. How do we make it? This is the mother cell that you need to remember. Hemocytoblast. And hemocytoblast will become myeloid. This slide is very important to remember because it's giving you an idea about everything. What's a mother cell and what are the stem cells, and each one will give you what. This is important. Okay? This slide is also very important. You need to know what exactly do we need to form the red blood cells. It's not only iron. It's all of this. If you have pernicious anemia, it's low red blood cells specifically because of vitamin B12. Pernicious anemia. Okay, and I talked about this, and I said we do not produce the intrinsic factor from the stomach for some reason. Pernicious anemia. Erythropoietin, this is important. Erythropoietin is the hormone secreted from kidney to stimulate the bone marrow to produce red blood cells as a reaction to hypoxia. What's hypoxia? Hypoxia. Low oxygen. Low oxygen in the blood. Blood types. We need to know this. What type A means regarding antigen antibody. B, AB, O. So this slide is important. This is what we have to know in this part, plus the RH. What's the antigen for RH? What is it called? D. Yes. So this is what we need to know in this part. And cross-reaction is also important, which is called a transfusion reaction. What does it mean? Incompatible blood leading to hemolysis. Cross-reaction. The next type is the white blood cells. General function. The number is important, 6 to 9. And how they work, the white blood cells. There are different types of white blood cells that we need to know what's the main thing about each one of those. Okay? So neutrophil, what do we need to know about neutrophil? Bacteria. It fights bacteria and it will make macrophages. Okay? Eosinophils, parasites. So if you have high eosinophils, you have parasites, eosinophils, and allergy as well. So there are like 1 or 2 words about each one of those. Neutrophils, bacteria, macrophages. It's called macrophages. They will make macrophages. Eosinophils, 2 words. Parasite, allergy. Eosinophils, parasite, and allergy. So if you have high eosinophil, if you do blood test or blood analysis and you see high eosinophils, it means 1 out of 2. You either have a parasitic infection or you have some sort of allergy. Allergy, okay? How about the basophils? Also allergy. Basophils, allergy, okay? The basophils and also the mast cells will produce the 2 H's. You need to understand what are the 2 H's that are produced and what's the function of each one of those. Histamine versus heparin. Okay? Monocytes, what's the importance of the monocytes? They will make macrophages. Neutrophils, micro. Monocytes, macro. The large eaters. How about the lymphocytes? Lymphocytes, generally speaking, specific defense. We have 3 types, T, B, and natural killer. What's the importance of each one of those? T, cell-mediated. They will attack directly. B, humoral or antibody-mediated. They do not attack directly. They release antibodies, and they have to differentiate into plasma cells first. Natural killer will kill your natural cells if it is cancerous or virally infected. And this is important for the differential count. So you should understand. If you have high neutrophils, what does it mean? High eosinophils, high mast cells, and so on. Those 3 terms are really important, and you will see it in the important concepts that's summarizing all of this. But you need to remember, what does it mean? Leukopenia, leukocytosis, and leukemia. So we talked about this already. Colony-stimulating factors, we have different types. You need to remember this. The M, the G, the GM, which is combined, and the multi. The next one is the platelets. What's the function of the platelets? Generally speaking, blood clot or coagulation, blood clot. So-called thrombocytes do not have nucleus. Okay? Lifespan, 9 to 12. It will be removed by spleen. And we only have third available, the rest is stored. What's a normal number? You need to remember this number as well. 150,000 to 500,000. If it is low, thrombocytopenia. If it is high, thrombocytosis. We make the platelets from megakaryocytes. Megakaryocytes break down into platelets. So megakaryocytes is like the mother cell of the platelets. You break it down to make platelets. TPO, interleukin-6, and multi-CSF, these are the hormonal factors that regulate the process of production of platelets. Hemostasis, vascular phase, platelet phase, coagulation phase. Okay, the vascular phase, spasm. Platelet phase, 15 seconds. And it's platelet adhesion and platelet plug. Platelet adhesion, platelet plug, which is a temporary seal. Platelets will release ADP, thromboxane A2, and serotonin, and this will start the process. And on the other hand, prostacyclin is released from the endothelial cells. The next phase is coagulation process, so it will start after the platelets, after 30 seconds, and it's basically fibrinogen to fibrin. You're forming blood clot. What is the blood clot? It is platelets entangled with fibrin. The cascade. The cascade, I mentioned that you need to know, generally speaking, extrinsic, how does it start? And it will end at factor 10. Intrinsic, how does it start? It will also end at factor 10. And then the common pathway. Common pathway is very important. Whether you start from extrinsic or intrinsic, you will go to the common pathway anyway. Whatever the reason is, you will end up activating factor 10, whether it's extrinsic or intrinsic. Factor 10 will form prothrombinase, which will convert prothrombin to thrombin. Thrombin will convert fibrinogen to fibrin. Fibrin will entangle the platelets, making the platelet clot. Bleeding time, normal. You need to remember this number, 1 to 4 minutes. And these factors are also important. What are the anticoagulants? Like antithrombin 3, heparin. And what are the factors that are needed to coagulate? Protein C, prostacyclin, you need calcium, and you need the vitamin K. So any one of these factors, if it is not available, that will make abnormal clotting, delayed clotting, dysfunctional, a problem. If you have one of those lacking, if you have less vitamin K, less calcium, less protein C, prostacyclin, okay? Then clot retraction will occur, and after that, it will be removed. The crust will be removed by the process of fibrinolysis. You need to remember this. Thrombin and TPA will activate plasminogen. Plasminogen will become plasmin, and plasmin is the one that's going to digest the fibrin. Important concepts, extremely important. Important factors from all chapters. This is summarizing all the concepts from the 3 chapters, and this will be used in the match. Okay? And that's it for chapter 19.